So, hi, my name is Alice. I'm working with Professor Adam Rosebrock in developing a minimal footprint microplate spectrophotometer for the purposes of continuous optical density measurements. So a little bit of background. The rate of cell population growth is often measured in order to study the effects of varying environmental conditions. And so this has very powerful effects in areas of biological and medical research. So optical density is essentially the amount of light that is absorbed by a sample given by these two equations that you see here. So measurements using optical density is very advantageous because it's very rapid and non-destructive to the sample. And it is often used in microbial culture growth and often used to quantify parameters such as concentration, cell mass, and growth rate, all of which require a continuous measurements. Because changes happen over time. And in addition to microbial growth measurements, you can also measure optical density to quantify changes in chemical compositions in techniques using ELISAs, for example, which also require continuous measurements. And so there are many different standards of measurements that are currently being used, many different types of equipment, and so I've separated them into two different devices, two different categories, one being multi-purpose and the other being single-purpose. So multi-purpose devices such as a multi-mode reader, which is a spectrophotometer, can be used to measure things such as fluorescence, luminescence, and absorbance. And it can also be used to measure an entire microplate, which is 96 different wells, which makes it very high throughput. But the disadvantage is that it's very large and very expensive, and they're not environmentally regulated, so that environmental dependent experiments can't be run in them. And in addition, they're also not continuous, which means that you have to manually put them into the multi-mode reader every time you want a data point. So an alternative to the multi-mode reader is a TCAN, which is also multi-purpose in that it can read fluorescence, luminescence, and absorbance. And it is also measures an entire microplate. But it has the advantage of being environmentally regulated and being continuously measuring. But the downsides are, again, that it's very large and very expensive. In addition, it also requires a lot of maintenance because it's using electromechanical systems which wear down quickly over time, and especially in high humidity and high temperature environments. And so an alternative to these two devices are the single-purpose devices, such as the UltraSpec and the EloCheck. And they're great because they're relatively small and portable and a lot cheaper than the multi-mode devices, ranging from $1,000 to $20,000 for the multi-purpose devices. And so these devices have the disadvantage of being only able to measure one sample per device, so that it is very low throughput. And in addition, they also require very large sample sizes. Now, the UltraSpec is not able to measure continuously, nor is it environmentally regulated, while the EloCheck can be continuous and it can be environmentally regulated. But as you can see, the multi-purpose and the single-purpose have their advantages and disadvantages. And so in creating a device, we wanted to be able to satisfy the constraints and criteria that we've gathered from examining these different devices. And so we want our device to be able to continuously monitor data so that the experimenter doesn't have to continuously be present when the experiment is running. We want the device to be high throughput, to be very low cost, so around under $1,000. We want it to be environmentally controlled for different factors such as temperature, humidity, and agitation for those types of experiments which require them. We also want the device to be of a minimal size, for it to be low maintenance, and for it to be flexible enough and modular enough such that you can switch out the different components if you need at different measurements, different wavelengths for different types of experiments, and for easy changing through maintenance. And we also want the device to be able to measure using small sample sizes, as well as to have no adverse effect on the samples themselves. And so, as a result, our objective to address the problems with current standards of measurement is to be able to create a device that is continuously monitoring, high throughput, low cost, minimal footprint, and low maintenance. So an overview of our device is that it's built around a 96-well standard microplate that you can see here. So there are 96 different emitter and detector pairs. And so the emitter circuitry sits on the very top over the top of the 96-well plate, and the emitter collimator sits right under that over the 96-well plate. And this collimator limits the amount of light that passes through with each emitter such that it is limited to the one well that they're meant to go through. And so the detector circuitry sits on the bottom of the 96-well plate such that the light path travels vertically through the sample plate into towards the detector. And so this is all controlled by a microcontroller, and in addition, the data is stored on an SD card on board, and it is also able to communicate via Bluetooth to other wireless devices. So the advantages of this device is that you're able to continuously monitor optical density changes as they happen. And they also, because of the microplate format, you're able to read very high throughput. And in addition, it's also very low maintenance because you get rid of the electromechanical component of many of the different other standards of measurements that are out there. It is also a minimal footprint, which means that it is able to fit into existing environmental control chambers that are currently being utilized in labs, and it's also a very small size. Each of the samples require a very small sample size, between 100 to 250 microliters, which means that you can save up on expensive reagents that are required for different types of experiments. And in addition, it is very modular between different detector and emitter pairs, and which means that it is very low maintenance for different experimental needs. So for testing, in order to validate the device, the accuracy of the device, we grew sample E. coli cultures in a sample plate and continuously monitor it during the incubation time in a environmentally controlled chamber with regulated temperature and agitation. And so the standard microbial optical density measurements uses 595 nanometers to 600 nanometers of wavelength. And so in choosing the emitters that we're going to use, we chose a light emitting diode, which is an LED, and it was chosen because it is very low power and it's also very cheap, and they're also a very small size. And so the criteria for the optimal emitter was that it was looking at the luminous intensity, which is the brightness, the peak wavelength between 595 to 600 nanometers for microbial measurements, and the cost of each LED in order to lower the cost of our overall device. And so from these different criteria, we chose two main emitters that we were going to test with the microbial measurements. And same thing for the detectors. We looked at a vast number of micro photodiodes and looked for criteria such as spectral sensitivity, which is the sensitivity of the device at 600 nanometers, the size of the active area for each photodiode, the dark current, which is a measure of the inherent noise of each of the photodiodes, and the cost of each photodiode. And from these criteria, we chose two main detectors. And so for bacterial testing, there are three main different samples that we used. One being the control well, which has no bacteria in it, and it's just media, which means that you don't expect to see any growth over time. The 0.05 optical density bacteria, which you expect to see, which is the normal optical density that you start off with when incubating. And so you expect to see a very normal growth curve from that. And in addition, we have a quarter diluted bacteria of the 0.05 optical density sample, which means that you should be expecting to see a delayed growth curve by a time lag of the doubling time of bacteria. And so in preparing our sample plates, we prepared two identical plates, one for the device to monitor and the other for a spectrophotometer in order to determine whether the device is accurate in its measurements. So as you can see, this is the raw data that we gather from the device. And if you may notice, they don't start off at the range that you expect them to. And this is because of inherent variations between each emitter and each detector. and there are inherent differences in each of them, in addition to plate bottom transparency variances. And so this will cause the device to measure a different value for each of the optical densities that you expect it to. And so in order to get rid of this variance, we normalized each of the data points that we've gathered to the range of the individual sample that it has been looking at over the range of time. And after we've obtained the normalized data, we adjusted this data to optical density. And so in doing that, as you can see, we've obtained very nice normal-looking bacterial growth curves that you expect to see off of any other of the current standards of measurements. And so you can clearly see the three different samples that you expect, the very bottom being the control plate, and as you can see, they don't grow over time at all. And there is also the first growth curve, which is the 0.05 optical density bacteria, and the delayed growth curve of the quarter diluted samples. And so one of the ways that we can validate the accuracy of our device is by examining the doubling time. And so for normal bacteria, you expect a doubling time between 15 to 30 minutes, depending on the environmental conditions. And if you measure the doubling time of the samples that we've measured using the device, you see that it falls well within that range of expected doubling time, which means that, great, the device is working. And in addition, we can compare the optical density that we've gathered off the device and compare it to the control plate that we've measured using the spectrophotometer. And as you can see here, all of the data points fall fairly well within the expected range of variance in between. And it can be noted that because you're taking the control plate out of the incubator every once in a while, there should be variances in the data between the control and the device plate. And so from these experiments, we want to be able to take it to the next step, in that we want to be able to make a microplate standard prototype, because currently the prototype that we're working with is not to microplate standard. And this is because we're making everything in-house, and it's very difficult to make all the circuitry fit into the board within the microplate because we're making it by hand. And in addition, we want to be able to better the emitter and detector alignment, such that we minimize the amount of motion-associated noise that we currently see with the device. And so, in conclusion, the device that we've currently built is able to continuously measure optical density changes as they happen. It will be of a minimal footprint, such that it is able to fit in a regular incubator. It will be very high throughput and will require very low maintenance with minimal electromechanical systems. It is expected to have a very low cost of around under $1,000 easily. And in addition, it requires very small sample sizes, and it's very modular for any different type of experimental need. And that concludes my thesis presentation, so thank you. So one of the criteria that we can sort of relax in order to make the device a little bit better is the cost constraint. Because we're very well under the $1,000 range, we could probably use different types of emitters and detectors that are slightly more expensive, but give a better range of detection for our different samples. And so this is one of the criteria that we can lower a little bit, because currently devices are within the $1,000 to $20,000 range, which means that any device that is built within that area would be perfectly fine. So if I had 30 more days to work on my thesis, I would definitely work on improving the size, reducing the size of the device itself. I was starting to work on a bit of that towards the end, but I didn't really get a chance to finish it, so I would definitely start with that. If there was one thing that I could change to my approach to my thesis project, I would probably spend a little less time on the planning and a bit more time on the actual making of the device, because I spent a lot of time in making sure that there wasn't any errors in the device, which is great, but I think I spent a bit too much time on it, so that's what I would focus on.